What the heck is that? Okay, can somebody confirm you can hear me?
I can hear you.
Thank you. Before we begin, are there any questions? All right, before we begin, let me state that I can find out who is attending the Zoom very easily. All I need to do is, let's see, come to the Zoom screen and then look at the participants. Like there's 13 people logged on, that's counting myself. And I can also go to my YouTube website and get a list of the number of people who have seen the YouTube website. And I know that the Thursday, there was only one student who requested it, and I had forgotten to put it up. So that means a large number of students never saw the lecture or the lab for Thursday. And in this class, you are expected to participate by either signing into Zoom or watching the Zoom session later on YouTube. So just be warned that I can find out who has been, um, well, participating in the class. And it will become a problem when some of the projects come up, like people get confused on the unknown project. And then I take a look and I see that the student has never seen the lab where we discuss the unknown project. And I'm not terribly very sympathetic for students who don't attend the class. Any question about that? Obviously, you guys are attending the Zoom session, so this is not intended for you guys. I'm just warning you guys that I can find out who has been attending the class. Any question on that? All right, if not,
I have a question. Are you gonna put the Thursday lecture and lab up?
it's up.
Oh,
I don't know what's wrong with my computer, but as you can see, it's showing actually, you can't see it yet. I don't have that displayed. I took it into Clark, I think on Friday or Saturday. No, it was Friday. And they had looked at it, and I guess I should have pointed this one out directly, and that is the computer isn't showing the screen normally. Let me share my screen. Oh, this is different. Okay, don't want that. There we go. That's what I want. And how do I get rid of that? All right, when I'm sharing my screen, you'll notice that the background is dark and the print is lighter. And normally it's the reverse, where the background is white and the print is black. And I don't know why it's doing that. So you'll just have to live with me. It's an odd color. And today we're going to be finishing chapter 1, hopefully, and then beginning chapter 2. Do realize that this week you do have a practice quiz 00, which must be taken by 11:59 p.m. Wednesday night. The practice quiz will cover the material in chapter 1 in lab 0 and lab 1, as well as cover the syllabus. Any questions about the practice quiz? You are encouraged to take the practice quiz. You'll get extra credit for it. And let me see how many practice quizzes I've got. You'll get at least one extra credit point 4 correct answers. I'm talking about for the practice quiz. I think it's 0.1 extra credit point for each question. On to week 2. Yeah, there's 2 practice quizzes. Practice quiz 00A, which will give you short answer and essay questions. I don't know why I brought that up, but you're gonna have 2 attempts on this one, but this one is not timed because some of the questions are essay questions, but it must be completed by, oh, that's not right. 8 p.m. I'm going to change that to 11:59 p.m. Change this date too. So it must be completed by 11:59 p.m. April 17th. In fact, you have 24 hours to work on that one because it's not using Respondus Lockdown Browser.
I have a question.
There are only a few questions like this. Just let me finish this and then I'll answer your question. And most of them will be on the final where we'll be giving you some essay or short answer questions, but this will help you with those type of questions. Most of your questions will be multiple choice and will require Respondus Lockdown Browser. All right, what was your question?
Um, I think you already explained it.
Okay,
Thank you.
great. And then there's a practice quiz 00B. This one does require Respondus Lockdown Browser. It'll be multiple choice. Most of your, yeah, this one has the wrong time too. It must have been this term. Students requested a different time. I have to go look at that for all the quizzes. Oops, didn't mean to show you that. I have to check the times in all the quizzes. All the quizzes will be due 11:59 p.m. If they're not, let me know and I'll change the time. I have a feeling all of them for this quarter are at 8 p.m. and I'm going to change them to 11:59 p.m. unless there's a request for a different time.
Does that mean that they're open tonight at 11:59 p.m.?
No. Oh, wait a minute. Oh yeah, they're open at Tuesday at 11:59 p.m. and they must be completed by 11:59 p.m. on Wednesday. So yes, sorry, I'm not thinking correctly. Because yeah, they open 11:59 p.m. Tuesday night, and then they close 11:59 p.m. Wednesday night, meaning you have 24 hours to take the quiz. But realize the Respondus Lockdown Browser quiz will be a timed quiz. So once you begin it, it must be finished within the time frame. And let me see if I can read how long you have. You have 13 minutes to finish this quiz once you begin it. Any questions about that?
13 minutes, how many questions?
Let me stop the share and I'll tell you because I don't remember.
Okay.
It's probably 10, but let me look. Yeah, there are 10 questions. Get out of that and I'll share my screen. Ah, there's 14 people logged in now. One more person joined the class. All right. Any questions about any of that? So hopefully we'll start chapter 2 and get far along on chapter 2. There is a lab at the lab time. I'll talk about that in the lab. I think I got chapter 1 here. And I've got marked that we were here. Is this the last slide we had talked about? Francesco Redi in an experiment showing that spontaneous generation did not occur for large animals like maggots, but there was the debate that spontaneous generation could still occur among microbes. And that's mainly because when you had a tube of nutrient broth, without doing anything in it, microbes would grow in it. And then the media, which would be clear, would become turbid with growth. About this time, did we talk about this slide? I think we did. I mentioned that Antony van Leeuwenhoek discovered the first microbes. So I think we've already talked about him. Rudolf Virchow proposed the theory of biogenesis, meaning that he wasn't the first person to propose it, but he's the first person to write it up and state that living cells can only arise from pre-existing living cells, becoming known as the theory of biogenesis. Arguments continued about whether spontaneous generation could occur with microbes until 1861, when Louis Pasteur came up with an ingenious scientific experiment disproving spontaneous generation and thereby proving the theory or confirming the theory of biogenesis. First, Louis Pasteur showed that microorganisms are present in the air and they can fall out of the air and cause the growth in a tube that's not sterile. And then microbes can be destroyed by heat. Louis Pasteur also showed that. And his ingenious, famous swan neck flask experiment disproved the theory of biogenesis. And that is, he got a glass flask like this with a long neck, and then he heated the neck here and then bent it into a swan neck, meaning an S-type shape. And then he heated the media to kill off all the microbes in the inside the tube and in the media. And then he left it open to the outside so that the vital force could come in, and the media stayed clear. There was no growth in the flask. The reason being that microbes from the air could come down, but then they would stop right there, and gravity wouldn't allow the microbes to come up the flask and then come into the media. However, if he did take the media and then brought it down here and then brought it back, the microbes would grow in the media. Any question about any of that? So after Louis Pasteur's day, not even microbes could spontaneously generate. And remember, the larger organisms were disproved by spontaneous generation from Francesco Redi's experiment. So with Louis Pasteur's work, a lot of advancements in microbiology occurred. And this brought about the relationship between microbes and disease, which was later called the germ theory of disease, that microbes might cause disease. And of course, we know that microbes do cause disease now, but initially it was just a theory that microbes may cause disease. Also came out of this is immunity, that people have an immune system which can fight off microbes which cause disease. And then out of that came the work on antimicrobial drugs, drugs that we can use against microbes. Any question about any of that? All right, so in the 1850s, Pasteur developed the basic idea of aseptic technique. He did come up with a way of getting a pure culture. However, it was very difficult. He used liquid culture, and what he would do is he'd make different dilutions and then have 10 tubes of each dilution. And if only one of the tubes had growth in it, and 9 of the tubes had no growth in it, he concluded correctly that probably about one cell caused the growth in the one tube, and so therefore it would be a pure culture. And most of the time he was correct. It was a pure culture. However, that was a very difficult way of getting a pure culture. And Edward Koch came up with a much easier way of getting a pure culture in 1881. You don't need to know the times here. I'm just giving them sort of to present it. He came up with streaking for colony isolation on a solid media. And most of the colonies that arise, if there's an isolated colony, came from a single cell. Now, in reality, it's only about 70% of the colonies come from a single cell, but it was a way of getting a pure culture. And he discovered that if he streaked it out twice, meaning you streak it out one time, get an isolated colony, take the cells from that colony, streak it out on another plate, get isolated colonies, and then take it from cells that have gone through 2 colony isolation events, the chance of having a pure culture are greater than 95%. And this was a very easy way of getting pure cultures, streaking for isolated colonies on a solid media. About this time, somebody in his lab, Hess, came up with nutrient agar, a new solid medium for growing lots of microbes. And this, actually all of these techniques are used today. Nutrient agar is one of the general purpose medias that we talked about. And then about this time is when Winogradsky developed the concept that microbes are involved in the biogeochemical cycles, such as the nitrogen cycle and the sulfur cycle. And he was able to do this, as well as his assistant or complement, whatever you call it, Badering, because they studied microbes in communities of different microbes growing together. And most microbiologists study microbes as a pure culture. The reason why you do study microbes as a pure culture, and that is if something happens in an experiment where you get a result, you want to know that one species caused that result. And if you have several species growing in the tube and you get a positive result in that tube, you don't know which species, or all of them perhaps, caused that positive result. And you just don't know. It may be just one species, or it could be several of the species. And that's why microbiologists tend to study microbes in pure cultures. However, Winogradsky studied them in communities of mixed cultures, and you have to study the biogeochemical cycles as a mixture of microbes because more than one microbe, lots of them, are responsible for the biogeochemical cycle. Like the nitrogen cycle, each step in the process occurs because of a different microbe doing that step of the nitrogen cycle. Any questions about any of that? All right. Out of this work came the concepts of fermentation and pasteurization. Louis Pasteur showed that microbes are responsible for fermentation. Alcoholic fermentation is when a microbe converts sugar to alcohol, which is used in making beer and wine. The beer industry actually asked Louis Pasteur to find out why their beer was going bad. And Louis Pasteur discovered that other microbes were growing in the beer and spoiling the beer, producing acetic acid from the alcohol, fermenting the alcohol into acetic acid. And that too is a fermentation process, but it's not one that somebody who's making beer and wine want. And out of that work, he came up with pasteurization, a method of treating the beer by heating it to kill the spoilage bacteria, but not hot enough to evaporate all the alcohol in the beer or wine. And this he called pasteurization. I think somebody else called it that, actually, he didn't, after Louis Pasteur. And it's the application of a high heat for a short time used to kill off the spoilage bacteria, but not destroy the product like the beer or the wine. And it's used in a large variety of foods, mostly in milk. It does not sterilize the milk. It does prevent the spoilage bacteria from being alive in the milk, but there are other bacteria in the milk. And that's why pasteurized milk has a shelf life. Any question about any of that? With the concept of fermentation, that microbes can affect an organic material, people began to ask the question, microbes have similar relationships with plants and animals, and what might microbes cause disease? This concept became known as the germ theory of disease. Originally, diseases were believed to be caused by poisonous vapors, and someone breathed in the poisonous vapors and then got the disease. So when someone got cholera when they went walking through a swamp, it was believed that they were breathing in the poisonous vapors from the swamp. In reality, they breathed in the cholera, which happened to be in the swamp. Any question about any of that? There were different people, Semmelweis and Lister, who show that steps could be taken to prevent disease by doing something like washing your hands or using a chemical disinfectant. However, the first person to show that disease was actually caused by a microbe was Robert Koch, Dr. Koch, and he proved that the bacterium Bacillus anthracis causes the disease anthrax. And then he wrote up the steps that he used to prove that this bacteria can cause the disease anthrax. And this became, and he published it, and this became known as Koch's postulates. The steps you use to show that a given organism causes a given disease. And this concept is so important, we're going to talk about it at 3 different times in this class. And so you can imagine there will be quiz questions on this topic, Koch's postulates, as well as a final question, because we're going to cover it 3 times in this class. And this proved the germ theory of disease. A given bacterium caused a given disease. Any question about any of that? And Koch's postulates were printed and published in 1884. You don't need to know the year, but you should know that they're published and they're called Koch's postulates. The steps you show that a given microbe causes a given disease. Around this time, Edward Jenner came up with the first vaccination. He made a vaccine against smallpox. What he did was he inoculated people with cowpox, another virus, and he observed the person was protected against smallpox. He actually experimented on two children, two boys. One was his own son, giving them cowpox, and then later found that they never got smallpox when smallpox came into I think he was in a town or a city, I don't remember. And at the time, he didn't really have any proof. He simply went on his own intuition. And he didn't know that milkmaids who frequently got cowpox were immune to getting smallpox. And that's why he suspected it would work. However, the concept of vaccines were not understood why the cowpox virus was protecting against smallpox until Louis Pasteur's work. What he did was Louis Pasteur showed that the cowpox virus is related to the smallpox virus. And then when the immune system mounts a response to the cowpox, it also mounted a response to the smallpox. So that when you got cowpox, you became immune to getting smallpox. And after that day, people understood how you could make make vaccines and why they worked. And after his day, different people made vaccines. Louis Pasteur made a bunch of different vaccines. The first one he made was actually in the chicken industry. He came up with a vaccine to prevent, I think it was getting cancer that was being spread by a chicken virus. He also came up with the vaccine for rabies. The term vaccination is derived from the word vacca, which is a word for cow. For Edward Jenner's vaccine against smallpox using the cowpox virus. The protection one gets from a vaccine is called immunity. Any questions about any of that? All right. Out of all of this concept came the concept of chemotherapy, the treatment of disease with chemicals. A chemotherapeutic agent is a drug that can be used to treat a given disease. When it was first used, and it can be used this way today, but it usually isn't, a chemotherapeutic drug is a drug that is used against a disease. Nowadays, when we talk about a chemotherapeutic agent, we almost always mean a drug that is used against cancer. However, you can use the word chemotherapeutic drug to mean a drug used against other diseases, including infectious diseases. However, we tend to use the word antibiotic to mean a drug which inhibits or kills microbes. Now, an antibiotic is a chemical produced by a living organism that inhibits or kills other microbes. This is the textbook definition of an antibiotic. And so penicillin G would be, by this textbook definition, an antibiotic. However, in common usage, including common usage among doctors, the term antibiotic is any drug that can be used against a microbe. So penicillin G is an antibiotic in common usage, and then ampicillin and carboxycillin are also an antibiotic in common usage. But by the textbook definition, ampicillin and carboxycillins are not antibiotics because they are not produced, at least directly, by a living organism, meaning I think carboxyllin is man-made totally, synthesized by men in a lab. And then ampicillin is made by an organism as penicillin G, and then people chemically alter it to make it ampicillin. And so by the textbook definition, it's not an antibiotic, but by common usage, it is. When I use the term antibiotic, I will use the common usage of the term. The first drug that was actually found to be working against an infectious disease, which was actually quinine from the bark of the cinchona or cinchona tree. Either pronunciation is correct. And this drug was used to treat malaria. Paul Ehrlich came up with the concept of using drugs to treat disease, and he simply tested different chemicals. And in 1910, after about 500 chemicals, he found a synthetic arsenic drug, which was able to cure the disease syphilis in 1910. And he called this a magic bullet, a chemical that could be used to treat an infectious disease. Any question about that? Oh, the drug he found was salvarsan, a synthetic arsenic compound. When do you think quinine was first discovered in Europe to treat malaria? Anyone have a guess? Nobody's going to take a guess? Well, think about it. I'm not going to give this one. So Paul Ehrlich in 1910 came up with a chemotherapeutic agent to cure syphilis, but it wasn't until 1928 that the first antibiotic was discovered by Alexander Fleming. What Alexander Fleming did was he was a sloppy microbiologist, and he made a plate of bacteria, and it had a colony of mold that grew on the plate. And he noticed around the mold there was a ring where the microbes, the bacteria, did not grow. And Alexander Fleming suspected that something was coming out of the mold, preventing the growth of the bacteria. And he was correct, and he discovered actually penicillin G, the first antibiotic. Penicillin G was not the first antibiotic that was first clinically available, because Alexander Fleming in his day could not purify it, and all he had actually was media that he was growing the mold in, and it would work against the bacteria. And you couldn't inject media into a patient and then get that to cure the bacterial infection. The first antibiotic was sulfonamide, a sulfur drug, and this was a man-made drug, and so they could purify it very easily. And it became clinically available in 1935. Penicillin G became clinically available in the early 1940s. And I'll try and tell you the story when we have time about the first patients treated with penicillin G. Any questions about that? Anyone come up with when the first drug, quinine, was used to treat malaria? The first chemotherapeutic agent?
I'll just make a guess. 1820?
1820. That's a good guess, but no, you're
Okay.
way too late. It was actually in the mid 1600s. What happened was Jesuit missionaries noticed the natives of Peru, meaning the American Indians of Peru, were eating the bark of the cinchona tree, to treat the malaria, and they noticed it was an effective treatment. And so they sent the bark of the cinchona tree to Europe to treat malaria. And of course, they brought the record with them to Europe with it. And so the first chemotherapeutic agent was long before the first drug was found as a chemotherapeutic agent by Paul Ehrlich. All right, let's talk a little bit about the classification of organisms. Microbes were once classified as plants. If you remember, Carlos Linnaeus classified all organisms into plants or animals. And hence the term flora was used for microbes. If you ever hear me use the term microflora, that is an old term that is no longer used, but I was taught it when I took microbiology, and it was used for bacteria. The correct term is microbiota now. When we talk about the human normal microbiota, these are the microbes that are normally present in and on a person. And animals and plants are host to symbiotic microbial communities, their normal microbiota, and they're central to their welfare. It is correct to say the human normal microbiota or the cat normal microbiota because it's different. Like cats have as part of their normal microbiota Bartonella henselae, which generally does not cause disease in cats. But if a cat bites you, a person can get, or scratches you, can get the bacteria causing cat scratch fever. Sorry, I just blanked out on the name there. And Bartonella henselae is not part of the normal human microbiota. It does cause disease. Normal microbiota is usually found on or in the skin, the oral nasal cavity, the upper respiratory tract. Usually the lungs do not have normal microbiota. It's found in the digestive and tract and the urethra. The normal microbiota helps prevent the growth of pathogens. For one thing, they can compete for the site. And if, for example, a human pathogen were to get on my skin, hopefully you can see this, I have normal microbiota on my skin. And so the bacteria that's a pathogen may not get established because it's already, instead of falling on my skin, it's fallen on a microbe on my skin. The normal microbiota can also compete for resources. And if the normal microbiota is used up all the resources, then the human pathogen has no resources, and then it can't grow and then cause disease. The normal microbiota, some of them can also secrete toxins that inhibit the growth of certain pathogens. We'll talk a little bit more about that later. And then some of the normal microbiota can produce nutrients and growth factors. For example, folic acid and vitamin K can be made by the normal microbiota, and a significant portion of the folic acid and vitamin K we get comes from our normal microbiota. The normal microbiota can also aid us in digesting our food. For example, we have E. coli in our gut, and E. coli helps us digest our food. Any question about any of that? The normal microbiota do not normally cause disease. However, they can cause disease in certain circumstances. One circumstance is if the patient is immunocompromised, then they're more likely to get a disease from their normal microbiota. Cancer patients, AIDS patients are two examples of people who are immunocompromised. An infectious disease is when a pathogen overcomes the host resistance, grows, and then becomes a pathogenic microbe causing disease. Any question about any of that? An emerging infectious disease is a new or changing disease that is increasing or has the potential to increase in incidence. Does anyone know of an emerging infectious disease? Come on, we're currently in a pandemic, and that's an emerging infectious disease.
COVID.
Yeah, COVID-19 is an emerging infectious disease. More than 4 years ago, I think it was 2019. So I guess that's almost 5 years, not quite 5 years, but was the first time COVID-19 hit people. So it is an emerging infectious disease, and eventually it'll just become a, what do you call it, a normal human disease, I guess, meaning once sooner or later, it'll be changed from an emerging infectious disease to just a, what do you call it, a normal disease. There is a term, but I'm drawing a blank on what it's called, an endemic disease, always present in the human population. One emerging infectious disease which they're watching is bird flu. There is concern that the virus, which spreads among birds, can infect people, and people can get sick, and in fact, it can kill people. But the only way people can get it is people who are touching or in close contact with birds. It does not spread person to person, which is a good thing. But they have noticed that bird flu is changing, and it now can infect mammals, and it can spread from mammal to mammal. I don't think they know exactly how it's spreading, but it's not spreading airborne, meaning I think it's cows who are getting it. And it's like getting into the cow feed. I don't know how it gets out of the cow and then gets into the feed, but then it can spread. Or it's getting on the milking device. And then when the farmer takes the milk device from one cow to another, he's spreading it among at least the milk cows. Something like that. The point is, bird flu could potentially spread among people if the virus changes. So they're watching it, and it's called the new and emerging infectious disease. It may have the potential to increase in incidence. So far, it has not, and hopefully it won't. It has not been airborne. How it spreads among birds is it causes flu among birds, and then the birds poop it out, and they poop on food or water, and another bird consumes that, and then the bird gets the flu. All right, so that's an emerging infectious disease for this lesson. All you need to know is the definition of an emerging infectious disease. I have other slides discussing emerging infectious disease, but I actually never discuss them, and we're a little bit behind schedule, so we're not going to discuss it this time. And there won't be any questions on any of this topic except on what an emerging infectious disease is. Any question about any of that? All right, if not, let's move on to Chapter 2. As is normal, I give you a introductory slide listing the topic, Chapter 2, Chemical Principles, and I give you the major goals for this lesson, as well as a rough outline. So always know the terms of the lesson, like what's an organic and an inorganic compound, hydrocarbons, polar and nonpolar molecules, hydrogen bonds, acid, base, and pH. Realize this is just the major goals. If you want all of the objectives, go to the objectives and overview of Chapter 2, and I list all of the objectives. For Chapter 2 here. And then on Chapter 1 and 2, I have it broken into an overview and objectives, and then the Chapter 2 study guides. For the later chapters, I haven't broken them down yet. My goal is to do that. So it will just be the Chapter 3 study guide. And at the start, it'll have the objectives, and then it'll go into the notes for Chapter 3. Any question about any of that? While I'm here, I've now finished Lecture 1. So let me make the PowerPoint available to you. And if it's not available after I finish the lesson, just send me a reminder that if I could make the PowerPoint available to you. Oh, and another thing I should mention, if I don't get the lesson, the Zoom lesson up by a day, send me a reminder that I need to get the Zoom session posted on my YouTube website. I normally try to do it right after the lab, but I must have got distracted last Thursday. All right. Also in Chapter 2, know the 4 major biological molecules and be able to describe the relationship and the characteristics of the monomer and the polymers for these 4 major biological molecules. Describe the composition, structure, and function of the 4 major biological molecules and be able to give examples of each. What are the 4 major biological molecules? Come on. And all food that we got 3 of them. So 3 of them should be very easy. You just look on a box of cereal and it'll give you 3 of the 4 major biological molecules. What are they?
Protein, carbs,
Protein, carbohydrates,
lipids, and
what'd
sugar.
you say, lipids?
Yeah, and sugar.
All right, sugar is a carbohydrate, but good guess.
Amino acids?
I'm sorry?
Amino acids?
Yeah, that would fall under the protein category, but good guess. The 4th is not on the box of cereal or any food packaging that I've ever seen, and that is nucleic acids. Those are the 4 major biological molecules. And
So
under protein, you'd have amino acids. Under carbohydrates, you'd have sugars. And under nucleic acids, you would have nucleotides, for example. And what those are those are the monomers of the polymer, meaning the protein is the polymer. The amino acids are the monomers. And we'll talk about that. In this lesson, you should also know the scale of things. Let me show this up. So you should know that a person or an organism is larger than a chicken egg, which is larger than a frog egg. And the frog egg is about the smallest thing that our human eyes can see. There are a few things smaller, but not much. You then have the microbes. Most eukaryotic cells Not all, like a frog egg and a chicken egg, but most eukaryotic cell, all prokaryotic cells fall within the range you can see with a light microscope. And a eukaryotic cell is larger than a prokaryotic cell, which is larger than a virus, which falls, most viruses fall beneath the range you can see with a light microscope. You need an electron microscope to see a virus, at least most viruses. Smaller than a virus is a large molecule like a protein. Smaller than a protein is a small molecule like water. Smaller than a small molecule are atoms. Any question about any of that? And about the smallest thing we can see is an atom, which you need an electron microscope to see. You do not need to know the exact sizes of these items. And let me state there's a mistake right here. That says 1 micrometer, and there's 1 micrometer. That should be 1 millimeter right there. But you don't need to know the exact size. Any question about that? You should know the relative relationship, like a person is bigger than a chicken egg, which is bigger than a frog egg, etc. In this lesson, we'll talk about what are organic compounds and what are inorganic compounds. Organic compounds tend to be complex structurally. They tend to have carbon holding the organic compound together. And carbon's important because it's an atom that combines to 4 other atoms. And like I said, organic compounds tend to be held together by carbon, linking the organic compound together. Organic compounds are typically held mostly by chemically covalent bonds, covalent chemical bonds. There are some other bonds that are in organic compounds, like hydrogen bonds, but mostly they're held together by a covalent chemical bond. And rarely they have ionic bonds also, but mostly covalent chemical bonds. An organic chemical compound typically lacks carbon, doesn't always lack carbon, and it's simpler, not as complex as an organic compound. They are required by living organisms in order for the living organism to grow, to repair themselves, to maintain themselves, and for reproduction. Examples of some inorganic compounds include oxygen and carbon dioxide, and you note the carbon dioxide does have carbon in it, as well as water. Water is one of the most important inorganic compounds in a living organism. It is one of the most important as well as the most abundant, meaning all living organisms are about 70% water. So obviously it's the most abundant or molecule in an organism. It's also one of the most important. Any questions about that? Let's talk a little bit more about water. Water is a polar molecule. What does that mean? Water has a region which is partially negative charge and partially positive charge. What happens is in the covalent bond between the hydrogen and the oxygen, why is my mouse dying? The electrons of that bond spend more time with oxygen than they do with hydrogen, making the oxygen slightly negatively charged. Your textbook doesn't call it that. I think it calls it a partial charge. It is not a complete negative charge. Everybody got that? It's a partial charge. The hydrogen on the other hand, is partially positive because the electron isn't spending much time with the hydrogen, it's spending most of the time with the oxygen. And it's actually both of the electrons in this covalent bond. So we call that a polar molecule, where part of the molecule is positive, part of the molecule is negative. I'm talking about partially negative and partially positive, not a complete charge. And that is a polar covalent chemical bond. Any questions about any of that? That differs. Let me move on to another example. Here we go. That differs from an ionic bond, which has a complete positive charge on the sodium ions, meaning the electron has left the sodium and gone to the chloride. The chloride has a -1 charge. The sodium has a +1 charge. If it's just a 1, people just usually go a plus and a minus. If it's a 2, they'll go +2. Okay? But my point is, this is a complete positive charge. This is an ionic bond holding the sodium to the chloride. It's an electromagnetic attraction, and that's what we mean by an ionic bond. Any questions about any of that? You should have had this in Biology 160. Anyways, because of the polarity of water, the hydrogen bonds can form between the positive, partially positive hydrogen and the partially negative oxygen. In water, a water molecule can actually form hydrogen bonds with 4 other water molecules, where this oxygen can bind to this hydrogen as well as to this hydrogen, and this hydrogen can bond to that oxygen, and that hydrogen can bond to that oxygen. This is an electromagnetic attraction, and it's called the hydrogen bond because it happens between this hydrogen and this other molecule, which is usually oxygen, but it could be nitrogen. In this case, in water, it is oxygen. This hydrogen bond is a very weak bond, and in liquid water, this hydrogen bond breaks and reforms all the time. I don't know if you can see this. And I got water in there, and you can see that water takes different shapes because it's a liquid. And the reason why water is a liquid is because this hydrogen bond is breaking and reforming all the time. Why liquid water takes the shape of the container it is in. We can reduce the temperature and make this hydrogen bond never break. And we do that by freezing the water. And as you know, an ice cube never changes its shape. And that's because the hydrogen bonds are locked in place. They do not break. And it's simply that the water has a low temperature, and so the hydrogen bond doesn't break. Any questions about any of that? By the way, water has 2 chemical bonds, liquid water at least, and that is it has a polar covalent bond inside the water molecule between the hydrogen and the oxygen, and then it has a hydrogen bond between different water molecules. We're talking about liquid water. When we're talking about steam, the water molecules have so much energy that they never form the hydrogen bond. So there is no hydrogen bond between steam, and that's why steam is a gas. And then when you freeze the water, the hydrogen bond has enough energy so that hydrogen bond does not break. And that's why ice does not change its shape. Any question about any of that? How we're doing time-wise? Oh, we got time. Let's watch the little video on water. Well, if I can get this to work, there it goes. Showing you how water breaks its hydrogen bonds and then reforms hydrogen bonds. Any questions? Okay. Water, remember, is an organic compound, and water is a good solvent. It's a good solvent because all polar molecules and ions can dissolve in water. So other polar molecules like water, where they have a negative, partial negative charge and a partial positive charge, those molecules can dissolve in water. One example is sugar. Sugar is a polar molecule and it can dissolve in water. Ions are ionic compounds like sodium chloride, which form a connection between the positive charge and the negative charge. Remember, these are complete charges. And you have a salt crystal. It's held together just by the electromagnetic charge between the sodium and the ion. But if you put sodium chloride crystal in water, the water will come up to the sodium ion here, and the negative oxygen will come up around the positive sodium ion and then surround it and then pull it out of the crystal. And then we'll surround the positive sodium, having the negative oxygen next to the positive sodium. And you have to have several water molecules surrounding the positive sodium, because remember that oxygen is only a partial negative charge, not a complete negative charge. And then the same thing happens with the negative chloride ion. The water can come up to it. Come on, mouse. Water can come up to it, and it will orient so that the positive hydrogen is next to the negative chloride. And then you'll have lots of water molecules surrounding that negative chloride ion to bring the chloride ion out of the crystal and in solution. Any questions about that? The important thing to realize is that all polar molecules and all ionic molecules can dissolve in water. Why we say water is a good solvent. Now, there are other molecules which do not dissolve in water. Everybody knows one, and that's oil. Like when you make salad dressing and you shake it up, the oil and the water or the vinegar separate. And that's because the oil doesn't like being in the water. It does not dissolve in the water. And that's because oil is a non-polar molecule. Non-polar molecules do not dissolve in water because water is a polar molecule. Any questions about any of that? Oops, I mean to go there. Doesn't look like that link works. Ah, remember that's partial positive, partial negative. This is a complete one, that's a complete negative one. So polar molecules are hydrophilic. What does that mean? Literally, hydrophilic means lover of water. But what we mean is the two like to be together. Nonpolar molecules are repelled by water. We call them hydrophobic. Literally, this means fear of water. They don't really fear water, they just do not dissolve in it. So hydrophilic, the substance dissolves in the water. Hydrophobic, it does not dissolve in the water. A substance which is hydrophobic is fats and oils. A substance which is hydrophilic, I've already mentioned, sugar is a hydrophilic substance. Acetic acid, like vinegar, hydrophilic. Trying to think of a few other, and I don't remember, other hydrophilic substances, anything that's ionic or polar. Hydrogen bonding between water molecules makes water excellent at buffering the temperature. You should know this because Western Washington and Western Oregon is buffered by the Pacific Ocean, meaning our temperature is buffered by the water of the Pacific Ocean. Water absorbs a lot of heat before it becomes heated and evaporates. And I guess as a comparison, Eastern Washington and Eastern Oregon are not buffered by the Pacific Ocean. And their temperature gets a lot hotter in the summer, and they get a lot cooler in the winter. And the reason for that is that their temperature is not buffered by the Pacific Ocean. Whereas in Western Oregon and Western Washington, the temperature is buffered by the Pacific Ocean. How that buffering happens is that it takes energy to break the hydrogen bonds in water. So a water molecule can take the energy of heat to break that hydrogen bond. The second thing that allows the energy to be absorbed is that the molecule of water itself can absorb the energy. All molecules can absorb the energy, and that we call the temperature of the molecule. What the temperature is just a measure of how much the molecule is vibrating and moving around. And so water, which is frozen, obviously is not moving very much. And that's why it is cooler in temperature than liquid water at, let's say, 4 degrees Celsius, which has some energy. And then as you warm the water up from 4 degrees Celsius, it still is a liquid because the hydrogen bonds can break, but they can reform. And as you warm the water up until you get to 100 degrees Celsius, when you get 100 degrees Celsius, you cause the water molecule to break the hydrogen bond, and the water molecule is now moving at a speed where it will not reform the hydrogen bond. And that we call steam. And that's why water at 100 degrees or higher is a gas, and below 100 degrees up until 0 degrees, it's liquid water, because the hydrogen bond will form, although it breaks, and that's why it's a liquid. And then below 0, the hydrogen bond does not break, and that's why it's a solid. Any question about any of that? All substances, or all atoms and molecules, can absorb the energy, and that will be the temperature of the molecule or the atom. And that's simply a measure of how fast that atom or molecule is moving and vibrating. So you could do that like on a rock. You can heat up the rock and the rock will get warmer. And you can heat it further and the rock will get hotter. It would not become a liquid until you applied a very, very high temperature, but you could apply a high temperature and the rock would become molten rock. Let's call it lava. And you could even heat it further, and then you would have, I don't know, gaseous rock. Any questions about any of that? Obviously, that would be a very high temperature. Don't ask me what it would be, but very high to be gaseous rock. Maybe not the temperature you find even on the planet Earth. Maybe you have to go to the sun to get that temperature. The point is, all molecules can absorb energy in the molecule and be the temperature of that molecule. The temperature is just a measure of how much that molecule is moving and vibrating. Water can absorb the energy into the temperature of the molecule. However, water can also absorb the energy by the hydrogen bonds, and that's why water is excellent at buffering the temperature, because it has two things that can absorb that energy. And therefore, if you have like a cup of oil and a cup of water, if you give the same amount of energy to the water as you do to the oil, the water will have the temperature raised less than the oil. Because in the oil, the only way that the energy can be absorbed is by the molecules of oil absorbing the energy, and that will be the temperature of the oil. Whereas in the water, both the molecule of the water as well as the hydrogen bonds can absorb that energy. And so the temperature of the water will raise less than the temperature of the oil. Any question about any of that? As hydrogen bonds are broken, the water molecule will evaporate, and this will take away the water molecule which has the highest energy. And therefore, water is good at cooling a person when they're perspiring or sweating. And that's because the hottest water molecule is the one that's leaving the person. The cooler water molecules remain with the person. And that takes the heat away from the person. Any question about any of that? All right. On the planet Earth, water can split into hydrogen ions and OH minus. Hydrogen ion is a proton. The OH minus is often called the hydroxide ion. On the planet Earth, that is all water does, unless you put electricity in it. And we're not going to talk about putting electricity in it, but you can, if you were to put electricity in the water, you can on the planet Earth split the water, meaning H2O, into hydrogen ions and OH minus ions. That does not happen on the planet of the Earth without adding electricity, where you split water into hydrogen and oxygen atoms. It does happen in the surface of the sun, where the temperature is hot enough that water can be split into hydrogen ions and oxygen ions. However, on the planet Earth, we don't have that high temperature, and then water will only split into hydrogen ions and hydroxide ion, the OH minus. When water breaks apart, that makes charged molecules, the hydrogen ion and the OH minus ion, and the water, which can help the water participate in chemical reactions. It also gives the water its pH. Any question about any of that? So if I ever ask a question, on the planet Earth, water breaks apart. Remember, it breaks apart into H plus ions and OH minus ions on the planet Earth. All right. When we're talking about the pH, we're seeing the pH scale here. Let me blow that up. If hydrogen ions are predominant over the OH minus, meaning there's more hydrogen ions than OH minus ions, it is acidic, and the pH is below pH 7, between 0 and pH 7. Increasing acidity the closer you go to pH 0. And that's increasing hydrogen ions in solution. Neutral water is when the hydrogen ion in solution equals the OH minus ions in solution. So pH water, pH 7 is neutral water, where the hydrogen ions equal the OH minus ions. And when you first have water, it'll be pH 7. The H plus will be equal to the OH minus. However, water will not stay pH 7, and that's because carbon dioxide will dissolve in the water, and carbon dioxide can form carbonic acid, or carbon dioxide reacts with water, making H2CO3. H2CO3 can break off the hydrogen, releasing hydrogen in solution. And so the carbon dioxide, when it dissolves in water, will become acidic. I don't have a good example of that, but most water, when it's been sitting in the air, will be around pH 5.5. Whenever the OH minus is higher in concentration than the H plus ions, we will have a basic solution, and the higher the OH concentration and the lower the H plus will be increasing basicity, until you get pH 14, where you'll have only OH minus and no H plus, and that's pH 14, which lime water is about that pH. Some things, seawater is slightly basic, or this is called alkaline also, pH above 8. I don't remember, it's 8.4 or 8.5. It's because of the minerals in ocean water or seawater, making the water basic. Milk of magnesia above pH 10. Also ammonia above pH 11. Also bleach is actually above pH 12. And that's probably the last, well, I guess you guys know an oven cleaner. That's actually the most basic substance most people are familiar with, an oven cleaner. That's above pH 13. Very basic. Human blood is slightly basic. I believe it's between pH 7.3 and 7.4. Milk, slightly acidic, a little less than pH 7. Urine, around pH 6. Wine and tomato juice, pH 4. Grapefruit juice, around pH 3. Lemon juice and vinegar, around pH 2. And then maybe the last thing you know, stomach acid, around pH 1.5. You guys might know something else, and that is the acid in your car battery is around pH 1. Any question about any of that? The stomach acid would be only when your stomach is producing the acid, and the pH will depend on what you've eaten, because the food can neutralize the acid. How are we doing time-wise? Oops, you guys have to tell me when I'm going past. All right, so let's end it here. Are there any questions? Where do we have that? All right, if there's no questions, I'm going to end it here, and I'll see you a little after 6:30. Let's make it 6:32 for the lab.